So hello everyone. So there was a technical problem, so I'm going to start introducing Golnaz Vahedi again. So she's an associate professor at University of Pennsylvania. She's from Iran and she did there her electrical engineer training. And as you will see, her lab is very interdisciplinary, which really has built up from this initial background she developed. So from Iran, Golnaz moved to Canada to complete her engineering training and then to the US, to Texas, where she did her PhD. And by then she felt that electrical engineering research field was already quite advanced and she looked for another discipline to follow her research career, and this is what brought her to biology. So her biological career started at NIH when she started as a postdoctoral fellow in the lab of John O'Shea, studying the epigenomic regulation of T lymphocytes in 2009. And a few years later, Golnaz was recruited as a young PI at UPenn. There, her contribution to the field of T-cell epigenetic and regulation of gene expression have been numerous and very important. So, for example, in collaboration with John Wherry, she has highlighted the key role of the transcription factor TCF1 in T-cell development, and further show that this transcription factor mediates this effect by promoting the interaction of specific topologically associated chromatin regions. She has also developed new methods for the analysis of single-cell transcriptomics and epigenomics, which she's using to study the regulation of gene expression in T cells in normal but also pathological contexts such as autoimmune diseases and cancer. So from a career perspective, Golnaz is the recipient of a number of awards. I will mention only some of them: the NIH Directors Award, which she obtained twice, the NAID Career Transition Awards, the Chan Zuckerberg Initiative Award, and the Michael Brown New Investigator Research Award. So today she's the Deputy Director of Innovation and Technology and Associate Professor of Genetics at the Perelman School of Medicine. And she's the co-director of the Epigenetics Institute at UPenn. So we look forward to Golnaz's seminar, but before, I must ask her the special introductory question, which in her case will be, if you could mentor your younger self, what would you say? Go ahead, Golnaz.
Well, first of all, Anna Maria, thank you so much for the introduction. And I'm sorry you had to do this more than once. I'm really excited to be here and share what we have done in the lab. But the answer to the question of what would I tell to younger self, it would be to invest more on interactions and people. So something that I realize, not that I haven't, but I think that's number one aspect of our work as a scientist, to build relationships. And maybe there's always a concern, or we are always trying to look for the next paper, the next grants, and there's always the milestones in our career that is driving us to move forward. But when I look back, I kind of see the most important part of my career has been the people that I interacted with when I was being trained with my mentors, and then now that I have my own lab, people in my lab, people that I train or interact with, how important that is, and how that's the number one investment of the career, not just for the sake of networking and being able to have more wins, but more of like how critical it is to build relationships and keep relationships and maintain relationships. So I would say I don't think I've done that as much as I want, as much as I should have when I was younger. And I hope moving forward I can build on this, and maybe people who are in this call, I will have the opportunity to interact more in future if I already am not connected.
Thank you, Golnaz, and let's hope for more interaction. And for now, just if you can share your screen. Yes, go ahead.
Absolutely. So you are able to see
yes.
my slides.
Yes. It's all good.
And it advances.
Thank you.
Fantastic. Thanks again. I'm really excited to be here, Anna Maria. Very great, generous introduction. Candy and all the global IMMUNOTALK organizers, Carla, Elena, and others. And I want to talk to you about kind of an overview of my lab, but with a very specific focus on an unpublished work in the beginning of my talk, where I want to start by kind of thinking about 500 million years of evolution and how the multicellular life first emerged from unicellular life. At this point of time, nature made a gamble, and this gamble was to duplicate the genetic material of the unicellular organism, as is being said, twice through the process of whole genome duplication, leading to multiple copies of the same genes that were working in unicellular organisms, and over the process of evolution, acquiring mutations among genes such that the same genes from the same family can now taking control of different cell types and leading to multicellular organisms and a specialized cell type and tissues. So this was such a good idea that nature kept doing it over and over. And of course, the process and the style of duplication have, or the kind of duplications vary across different genetic material. We have numerous cases of tandem duplications, segmental duplications, and also when a piece of DNA is really critical and is functionally important, through the process of retrotransposition, that piece can be duplicated on a completely different part of the chromosome. And again, over years and years and years of evolution and selection, the DNA acquire mutations, and only the combination of genes and mutated genetic material that are passing the process of selection and beneficial for the host remain in the genome. So I've been interested in these concepts as a broad genetic event, and moving millions of years into the mammalian genome. A very simple question we asked in our lab was, which our lab is very much, as Anna Maria mentioned, is interested in studying transcription factors in the context of T cells. We asked a very simple question. So how different transcription factors from the same family, which they are referred as paralogous transcription factors, where are they located? Are they typically happening completely isolated from each other on different chromosomes, or they happen to be located side by side and maybe taking advantage of genomic proximity to each other? So if you look at the mammalian genome, I'm showing you some numbers for the human genome, but that is also true for mice. In fact, majority of paralogous transcription factors, meaning transcription factors from the same family, where they belong to that family based on the conservation of their DNA binding domain, but they are, as I mentioned, they are completely different. Typically, they play different roles across different tissues, although some also play conserved roles, including MYC working for proliferation across many different contexts. But when you look at all existing paralogous transcription factors and ask from the same family, where are they located? Majority, 90% of transcription factors are isolated from the other members of that family. For example, on a chromosome that MYC is located, you cannot find any other transcription factor from the HLH family of transcription factors. However, there are exceptions. About 10% of the genome, some clustering of paralogous transcription factors happen, meaning that transcription factor genetic material is proximal on the DNA and side by side. About 5% of the paralogous transcription factors plus have represent this clustering of more than two. For example, the stat locus that I was very interested during my postdoctoral work in the O'Shea lab, it represents this level of clustering, the stat3 and stat5 and stat5e, B, where these stat proteins have very 90%, 95% similarity on their DNA binding domain and their recognition side on DNA, yet they are doing really distinct functions. They are clustered side by side. And the part that was interesting to us was that the same kind of frequency of events also happens pairing transcription factors from the same family, meaning two transcription factors, for example, 6.2 and 6.3, are located side by side. And if you think about it, this pairing seems to be a quite high frequencies because the number of clusters could be quite variable. So you can have 3, 4, 5, etc. In fact, there are interesting transcription factors with a large number of clusterings, including HOX-A cluster, which is about 12 transcription factors that are located side by side. HOX-A is important for patterning and embryogenesis. Another very large cluster of paralogous transcription factors actually is the crab-zinc finger proteins that are among the fastest evolving transcription factors. And in fact, they are very different across different individuals and mammalian genomes. So a very broad question that my lab is interested in is relatively actually a new direction for us. And I don't think today I'm going to answer this. Maybe we spend many decades remaining in my career to try to better understand and answer this question is whether genomic pairing of these transcription factors from the same family provides any evolutionary advantage. These 5% of transcription factors that I mentioned to you that through the process of selection over millions of years happen to stay together side by side, whether there was a reason for the host to actually keep two transcription factors from the same family together. So the transcription factor family I want to talk to you today, and we started and focused to answer this question, is the ETS family proteins. ETS family proteins actually, again, started in multicellular organisms, is very ancient and dates back to 500 million years ago. And in fact, you find ETS1, which is the ancestral gene, in the sponges. There are about 28 different ETS proteins in vertebrates, and Fly1 is one of the important transcription factors in vertebrates, which occurred and is important for vascularization. So ETS transcription factors are quite complex, and immunologists have been studying ETS proteins for many decades. People in the call or listening to this talk, it is very likely that you've encountered these proteins or you're studying them or in different contexts. I'm sure you've heard of the PU.1, which is important for SPI1, important for myeloids and B cells. ETS1, as I mentioned, is the ancestral gene that every other duplication actually started and dates back to ETS1, the original transcription factor, which is been documented extensively to be important for T cells and K cells. If you don't have ETS1, T cell development is affected and NK cells are affected. And many other of these transcription factors either have known functions in the context of the immune system or are being implicated in immune system or other systems through mostly the knowledge of mouse deletion strategies. You completely remove or perturb genetic context of these specific genes one at a time, assess the immune function, and report that one or more than one program is affected. Going back to this concept of genomic pairing, what we were interested was whether its family have any report of genomic pairing that are of interest and for investigation for us. So if you look at the 28 transcription factors, about we are able to find 4 its family pairs in the mammalian genome. The first one, again, because I focus on this because it's also the ancestral gene that all the duplications that started from is ETS1. Happens at this locus in the human genome on chromosome 11, where you have 2 ETS transcription factors, ETS1 and FLY1, about 200 kb apart and antisense to each other. Quite interestingly, we have another paralogous ETS transcription factor positioning of ETS2 and ERG on chromosome 21. Again, very similar positioning, 200 kb antisense. And in fact, when you really perform sequence alignment for proteins, and maybe in the previous slide, I also have some assessment of this, ETS1 and ETS2 are the most similar two transcription factors in the ETS family, meaning that ETS2 probably arose from ETS1. ERG and FLY1 are the most similar. In fact, there are some flanking genes here, KC and G, that also have quite similarity. And we reason that perhaps what happened, again, millions of years ago, ETS1 made through the process of tandem duplication gave rise to FLY1. And this kind of pairing was such a good idea that through the process of retrotransposition, this whole segment, maybe a megabase pair or longer piece of genome, was through the process of transposition, got duplicated on a completely different chromosome. There are other pairing of its transcription factors, EHF, ELF5, ETV3 and ETV also that happen in the mammalian in the human genome. So if you look at where these transcription factors are expressed, just very briefly using some existing single cell data, what you can see is a very striking pattern or some sort of a coupling of these paired transcription factors. First of all, ETS1, FLY1 are the most expressed in the context of T cells. The other pairs are expressed, but not necessarily to the same extent. And the very obvious pattern is that ETS1 level is very high in T and NK cell, and that's much higher than FLY1. But again, FLY1 is expressed to some extent. ETS1, FLY1 are also expressed in the context of B cell and a slightly different proportion. And ETS2 seems to be the ETS2 ERG seem to be the kind of a paired gene with acting similar to ETS1, FLY1 in the context of myeloids. So we are a T cell lab, so this striking pattern of ETS1 expression level was a reason for us to focus on this pair to better understand whether there is something about the genomic pairing of these two transcription factors that can tell us about their selection and evolutionary advantage of being positioned together. Another reason for us to focus on this locus was the fact that if you look in the human genome and superimpose the genome-wide association studies information in complex immune disorders, the striking observation was that there is no complex immune disorder that it doesn't have a risk variant associated with this locus. So these are single nucleotide polymorphisms associated with a variety of these complex disorders that I listed here. And this enrichment suggests that perhaps this locus is very sensitive to genetic perturbation and for the immune system and immune function. So kind of refining our question, we want to understand how genomic pairing of S1 FLY1 provides an evolutionary advantage by shaping how these two genes are regulated at the same time in the same cell through the lens of chromatin architecture, because that's the usage of DNA and utilizing the genetic material is dictated through the chromatin organization. Maybe just a primer for how the chromatin is organized inside the nucleus. We just wrote, two PhD students in my lab recently wrote a review article that you may be interested in taking a look. Just for the sake of this talk, DNA is wrapped around these spools called nucleosomes, where they contain 8 histone proteins. The histone tail can get modified, and depending the chemical modification, it represents openness or closeness of the DNA. For example, if lysine 27 on histone 3 gets acetylated, the chromatin is more expanded and there are more accessible regions, and this has been shown be the signature for active enhancers. If the histone tail on the same lysine, on the same histone get trimethylated, it's a sign for inactive or repressed chromatin. And again, gene regulation happens by regulatory elements called enhancers being in spatial proximity to their target genes and leading to transcriptional firing of those genes. And then the broader scale DNA is packed into these structures called topologically associating domains or compartments. So the focus of our lab was to better understand genome organization in this locus. The kind of classic genomic assay that you measure through the genome interaction is Hi-C, where the experiment ligates different pieces of DNA that are in spatial proximity in the 3D space. For example, if I have a signal here, it means that this piece of DNA and this piece of DNA were interacting in the 3D space and forming a loop. So by just looking at this locus and the Hi-C measurement in thymocytes, which is going to be the cell type I use for the main part of this talk, what you can see is that this 1 megabase per genomic locus is segmented into kind of 3 different domains. This domain that seems that there are a lot of interactions between different components of DNA harboring the FLY1 gene. This large domain that has ETS1 and this stretch of enhancer elements that is referred to as super enhancer also is annotated as a long non-coding RNA, which I'm going to be using in the rest of the talk, and also this other small domain. And there are some interactions between these domains. However, the blackness of this component of the heat map suggests that ETS1, there is an insulation or some sort of a separation between ETS1 and FLY1 in the T cells genome. Another important point to note is that the gene body for ETS1 is highly acetylated, and also the gene body for FLY1 to some extent are acetylated. And through the definition of stretch of enhancers, these regions also score as a super enhancer. Another important point is that these regions and the loops are stabilized by the binding of a structural protein called CTCF that you kind of see them at the boundaries of these domains. So to better understand the chromatin structure, we needed these tools we cannot go back to 500 million years of evolution, but we can start perturbing the genome and ask how the genome changed, and whether from those changes we can infer why these two genes are located side by side, and is there any advantage for their genomic positioning. So the first genetic perturbation we used CRISPR-Cas9 in collaboration with my colleague Jorge Mejia here at Penn, and we removed the super enhancer 25 kb stretch of enhancer elements that is completely separate from the ETS1, but we made it CRISPR mice where we deleted this piece of DNA in the entire genome, into all the cells in an animal, and asked, does it even matter for the mice not to have this stretch of DNA sequence? When we and the critical question that might be very important to first tackle is that did anything change in the context of the immune cell function? So this data is published. I move quickly, again, in collaboration with Jorge. We started to model different immune perturbations. And we started from the allergic inflammation, modeling allergic inflammation, mostly because this is stretch that we deleted actually had 12 SNPs associated with allergy and asthma in humans. So Michael, a talented postdoc fellow in Jorge's lab, modeled type 2 immune responses by through the house dust mite installation of intranasal installation of house dust mite extract through this scheme. And we were able to see a huge infiltration of eosinophil in the lung of the super enhancer knockout animals, and also correspondingly expansion of Th2 cells. So again, this piece of work is published. So I just want to summarize by in kind of the functional and in vivo assessment of this genetic perturbation. What we found is that this is a work from a talented postdoc at the time, who Aditi Chandra, who has her lab right now, another talented postdoc, Sora and Michael. We found that this very structured genome organization at the S1FLY1 locus is critical for controlling the amount and dosage of S1 level. And when you don't have this super enhancer, again, we're not creating an S1 knockout mice, just the amount of S1 mRNA and protein is reduced. But that is very critical for CD4 T helper differentiation and a balance between Th1 and Th2. That balance ultimately leads to allergic inflammation. Some ongoing work in the lab from a talented technician, Jemmy, is to show how, in fact, as these mice age, there are so many other perturbations in their immune system, including increase in the activated CD4 and CD8 T cells as the mice age. So we have also made some other perturbations, including removing the CTCF, and we have seen some defects in the B cell compartment. And these are kind of just, I'm showing and sharing these to show you the flavors of different directions that this locus is trying to take us. But I want to conclude here that B cell and T cell are indeed, by using mice as a model, are very sensitive to genetic perturbation in this locus, and perhaps that's why we still don't know the detailed mechanism for each and single one of these events, but that's why SNPs in this locus are highly associated with complex disorders. So in the remaining part of my talk, I just want to show you some unpublished work on how we tackle to answer the cis questions, meaning how does the transcripts of the cell change when we make these genetic perturbations in the wild type and in the after genetic perturbation? And how does the 3D genome rewires after this perturbation? And maybe this type of analysis, again, bring us back to potentially answering the benefits of these two genes being located side by side. So we got, to answer this question, we fully utilize or complemented our sequencing experiments with microscopy techniques, because you really want to be able to answer what happens to both of these genes in the same cell at the same time. So we performed the first set of experiments we did. We wanted to answer what is the transcription of these two genes inside the same cell, and we performed single molecule RNA fish. And we found that, in fact, majority of cells in thymocytes, T cells from the thymus, express high level of ETS1 and low level of FLY1, represented by kind of this specific example. However, there is just some minority of cells that the two genes are co-expressed. Again, if you think about neighboring gene, usually the most common idea is that they are together at the same locus, so they might be co-expressed because transcriptional machinery is trying to apply the same rules. We have that, but it's really the minority of T cells, and majority of T cells have ETS1 seem to have an advantage in its transcription compared to Fly1. And this is in line with the sequencing, single cell sequencing experiments I showed you in the beginning. But we wanted to see how does the chromatin fiber actually looks like inside the cells. And a very talented PhD student, Atisha J, implement in collaboration with my colleague Babak Faryabi, Yechao Zhao in his lab, and computational work Sora Yoon in my lab, implemented what is referred to as optical reconstruction of chromatin architecture, or ORCA, to reconstruct how the DNA fiber looks like inside every single cell. So the way this technique was developed is also called sequential FISH, and was developed by Alistair Bodeger. We got a lot of help from him and his lab. The way this technique works is kind of like DNA FISH. You design a probe for a segment of genome, and there are fluorophores that have homology to this region of interest, and you perform microscopy to register the XYZ of that region. In sequential labeling, the probes are designed for the region of interest. In this case, the 1 megabase per segment of our interest that harbors ETS1, Fly1, and the super enhancer. And we have a microfluidic attached to the microscope that perform this sequential labeling one at a time. So we apply reagents that correspond to DNA FISH for the first segment. We perform imaging, and we register the XYZ of that particular segment. The second very critical part is to apply reagents complementary to first set of probes to remove the original probes. We also take an image because we want to be sure that we cleared the signal. And then the microfluidic adds the reagents for the second sequence, and that goes on until we complete what is referred to as a walk along the chromosome. So using this process, we can reconstruct images like this, which are all coming from the microscope, and provide us with the information how this linear piece that you kind of draw, you can use your genome browser, looks like at the 3D space. What's the most critical part, and took us a lot of time, was to show how high quality our ORCA experiments are. When you in ORCA, you measure the actual distance in the 3D space of the nucleus. If you pool all the single alleles and look at the average distance and just create a heat map of the average distance for pairwise distances, you can see a very striking similarity between about just 1,000 or so alleles of ORCA coming from microscope and millions of cells coming from Hi-C. So the correlation is quite high. So we are satisfied with the quality of these data sets. But let me show you again, in the interest of time, I just want to show you some highlights of the kind of analyses that you can do across thousands of alleles and come up with a lot of comparisons in distributions. But I want to show you the upshot of this analysis in the wild type setting using one specific trace. So again, this is an actual trace from a DNA that came from this walking along the chromosome in the context of thymocytes. And the most clear observation presented by these distributions is that the ETS1 genomic element and its super enhancer are very frequently very proximal to each other, about 100 nanometer or so in the 3D space, and also represented by this distribution. And the Fly1 gene is a slightly separate and have a distance from this this gene and ETS1 and its super enhancer being proximal. This perhaps explain why ETS1 has a very high level of transcript in the context of wild type T cells. But then we wanted to understand what happens when the genome when we make a genetic perturbations and how the genome rewires after we remove the ETS1 super enhancer. This is, as I mentioned, a critical important part is that when we perform, when we design the probes, the way we deleted, we have a 25 kb that is deleted, and the flanking region still we have probes that we can assess the 3D genome positioning of the flanking probes within the proximity of this genetic perturbation. So the first experiment, we wanted to understand how gene expression changed after this genetic perturbation. As I talked to you about the wild type RNA fish before, we have ETS1 high cells, most of cells are ETS1 high, Fly1 low. And if you compare, calculate the Spearman correlation of ETS1, Fly1 across individual cells, they are not very highly correlated. There is some correlation, but it's not very high. When you remove the super enhancer, what really happens is that you kind of now have the co-expression of these two genes, meaning that in majority of cells, as represented by this high correlation, there is comparable amount of mRNA molecules that are being made from ETS1 and Fly1, and this exclusive behavior of ETS1 high cells is disappearing. But how does the DNA lead to this change in transcriptional firing? What ORCA shows us is that the DNA completely rewires and really positions Fly1 locus, which, if you remember, was highly acetylated and had a high stretch of enhancers itself. And the DNA rewiring happens such that Fly1 is now positioned very proximal to ETS1. And the RNA molecule that are being fired from the ETS1 locus is now moderate, with the consequence of more Fly1 transcript being made. And we think this might be the reason that, and in fact, the piece of DNA that harbored the super enhancer is very now far away, suggesting that if it's not acetylated, it no longer has that exclusive behavior with ETS1. And we reason that this might be because of the importance of ETS1 in T cell development and how this rewiring, in fact, enables cells to pass through T cell development, lead to no major defect in T cell development. However, the reduction in the ETS1 level will be consequential in the context of T helper differentiation, where high amount of mRNA molecule is required. And maybe that's why the allergic inflammation is the consequence of this inability of ETS1 level to be able to increase to the level that is needed. And maybe with this way of kind of simplifying or kind of summarizing the data I showed you so far, we don't have any evidence why these two genes are located side by side and what is the evolutionary advantage. But we are kind of postulating that maybe there are, because of the fact that these two transcription factors are from the same family, and the potentially protein products of these two transcription factors have some consequences on the nuclear space, maybe through phase separation and condensate formation. We think maybe the protein outcomes of the locus have some ability to reorganize and give a feedback to the locus and to the transcriptional machinery. And by being able to respond back to the DNA, the absence of the super enhancer that is critical for ETS1 level in the context of T cells, it is possible that the Fly1 protein and Fly1 genomic locus have some ability to sense the absence of the super enhancer that is critical for T cells, rewires the genome, and lead to no major defect in T cell development and potentially compensate for this absence of critical regulatory element. So maybe the positioning of two members of the same family has some advantages in this sense. And again, this is an active work for us. We are really in the beginning of this type of directions. And maybe through the Q&A, I will be looking forward to hearing your thoughts on what you think maybe might be some evolutionary advantage of these type of paralogous transcription factors to be positioned together. In the remaining few minutes, I just want to highlight some other work that are going on. I talked to you about the 25 kb. I showed you a lot of our data about this entire segment being deleted. We have narrowed down this 25 kb, in fact, to 4 kilobase pair, and there is only one SNP that is associated with that 4 kilobase pair in human allergy. We are working with at Penn Medicine Biobank and Immune Health to recall human individuals in the Penn medical system with and without the variants that I mentioned. And we want to better understand whether the mechanisms and chromatin rewiring that we identify in mice also happen in human samples. But the other piece that is equally, maybe even more so exciting for me and my lab at the moment is how interesting that another part of the genome has something very similar to X1-Fly1, the X2 and ERG locus, very similar arrangement, but they are they are totally not used in lymphocytes, although ERG has important roles in hematopoiesis and maybe some early stages of T cell development to maintain the stemness. But overall, this other kind of genomic pairing is very specialized in other systems, including myeloid. So we are really excited to better understand whether this common, what was about this genomic pairing that, again, nature really found it very useful to control such critical immune cell types in the mammalian genome. So this wraps up the first part of my talk. I promise not to go over time. I just want to give you some brief ideas about other directions that are going on in my lab. As I mentioned, we are interested in transcription factors. And Anna Maria mentioned how we have been working on TCF1, which is important for T cell development. And we showed how TCF1 is important for opening the chromatin, 3D long-range interactions. And the most recent work from a PhD student in my lab, who's now currently a postdoc in Arlene Schorf's lab at Harvard, try to focus on which part of TCF1 is important for T cell development. She looked at this intrinsically disordered feature of TCF1, but had a global look at creating large number of truncation mutation without having any bias. She used the OP9 delta-like 1 system and gene replacement strategy, use a TCF1 knockout and remove TCF1 replace TCF1 with these mutants, and found that in fact one of these truncation mutants led to a dramatic defect in T cell development. This particular piece is in fact intrinsically disordered experimentally and computationally. We also showed that. And what was quite nice was we were able to replace and rescue the phenotype by swapping the intrinsically disordered domain from a parallel transcription factor in B cell development, EBF1, and we're able to rescue, maybe not to the same extent of wild type cells, but this was quite remarkable for us, thinking that there's something interesting and shared between the intrinsically disordered domain. An ongoing work in my lab is try to narrow down what is it in the L1 domain that is really critical. A PhD student in my lab, Carlos Pandevida, is trying to better understand this. And he has, in fact, narrowed down this the critical component of L1 to 4 residues, and we are very excited to better understand the role of these 4 residues. Last but not least, I want to share some kind of profiling and global work where we are trying to understand how T cells contribute to autoimmunity. We're part of a program called Human Pancreas Analysis Program, or HPAP at Penn, led by Dr. Naji, where we work with human organ donors with and without type 1 diabetes and look at their pancreatic lymph node and other tissues. And I've been trying to do multi-omic experiments to better understand what are the differences in the immune cells in pancreatic lymph nodes of individuals with and without type 1 diabetes. This is a work from talented postdoc Mariam Abedi and Priyadarshini Rai. They generated the data and analyzed the data, lots of clusters, annotation. Just want to highlight one piece of very interesting observation that we found. We found a CD4 T cell subset that was very unique and only present in type 1 diabetes and autoantibody positive individuals. The characteristics of these unique cells is that they were annotated as central memory T cells. They don't really express cytokines. They have a really dramatic chromatin and gene signature of NF-kappa B and BOC2. It's in terms of expression and enrichment of these factors, motifs within ATAC-seq data. And most importantly, we were able to find these cells in the NOD mice at pre-symptomatic stages. Again, I would love to better understand the relevance of NF-kappa B in cells that do not express cytokines, behave like memory cells or stem-like cells. If you go to this literature of CD8 T cell, TCF1 high, CD8 T cells, there is almost every paper, the NF-kappa B gene expression is enriched, but nobody really has fully explored what this means, that you have a stem-like cell that it seems to have all these activation signatures. So again, I would love to hear from you if you know what NF-kappa B signaling is doing in this type of cells. And with that, I apologize, we went a little bit fast at the end, but I want to give you some ideas of the different kind of work we are doing in our lab. So I'm incredibly fortunate to work with talented trainees, the current members of the lab. I mentioned their names as we move forward, and the former trainees. We collaborate, and part of our success is because of our colleagues and collaborators being exceptional. Collaboration with Faryabi Lab, Arjun Raj, I don't know if I mentioned, he helped us with a lot of RNA fish experiments, but he also has a very exciting and very rigorous and easy tool to use to analyze RNA fish data. It's called Nimbus. You can just make an account. There is a free account and a paid version. I highly recommend it if you do RNA fish experiments because it really simplifies data analysis. Jorge Henao Mejia, John Weary, Ivan Millard, Norbert Purdy, Barbara Key gave us some X1 reagents, and my HPAP collaborators, and also the funders. And again, thank you so much. I'm sorry for the technical difficulty in the beginning, and I really look forward to more interactions with you all in coming years, and look forward to your comments.
So thank you, Golnaz, for this wonderful journey with transcription factors and chromatin. To all of you, please don't forget to ask your questions to Golnaz on the YouTube channels in social media, as indicated here on the slide. And let's meet again next week for a new talk. So next week we will host Michelle Xu for Tsinghua University in China. Thank you and have a beautiful week. Bye-bye.
Thank you. Bye.